Hi, my name is Fran Querdasi, and I'm a developmental psychology PhD student at UCLA, where I study neurobiological mechanisms that underlie links between early adversity and health. Today, I will be sharing some of my recent work exploring the relations among intergenerational adversity exposure, composition of the gut microbiota, and somatic and emotional health symptoms in early childhood. Recent research on developmental impacts of exposure to early adversity supports the idea that postnatal exposures, like childhood abuse or neglect, and prenatal exposures, like abnormal levels of glucocorticoids while in utero, can impact children's subsequent risk for psychopathology. Furthermore, exposures in the parent's generation, like a mother's own childhood maltreatment, can also impact children's risk. One mechanism that is thought to underlie the link between adversity and risk for psychopathology is the microorganisms called microbiota that live in the gastrointestinal tract or gut. Preclinical research and small studies in humans have linked preconception, prenatal, and postnatal adversity exposures with differences in composition of the gut microbiota. However, no studies have yet used large sample sizes to look at the shared and unique impacts of different adversities on gut microbiota composition and associations of the microbiota with child development, which was the aim of this current study. So for this study, we had a community sample of 450 mother-child dyads who participated in the longitudinal birth cohort study called Growing Up in Singapore Towards Healthy Outcomes, or GUSTO. We measured preconception adversity with mother's report of her own childhood maltreatment, prenatal adversity as mother's report of anxiety symptoms during pregnancy, and children's postnatal adversity exposure with children's exposure to stressful life events from birth to 2 years of age. Finally, child mental health in the form of internalizing symptoms and somatic health in the form of gastrointestinal distress were measured as outcomes. Children donated a stool sample at 2 years of age, from which we computed metrics of gut microbiota composition. So moving on to the results, as can be seen in the upper right panel of figure 1 here, we found differences in alpha diversity or within individual diversity as a function of postnatal stress only. On average, children exposed to postnatal adversity, so the dots in turquoise here, had bacterial species in their gut that were less diverse or more similar genetically to one another than children who are not exposed to postnatal adversity, the dots in red. The blue bars here represent the group means, and this finding is in line with prior work that found lower alpha diversity among children exposed to caregiving adversity at around 8 years of age. So moving on to figure 2, we found several taxa whose abundance significantly differed according to adversity exposure. On the x-axis, log twofold change captures the magnitude of the regression coefficient for each taxon, and names of the taxa are on the y-axis. Some of these findings are in line with prior research. For example, we found that taxa from the Lachnoclostridium genus are less abundant among children whose mothers reported greater childhood maltreatment. Another study also found lower abundance of taxa from the Lacnospiraceae family, which contains the genus Lachnoclostridium, among children exposed to early caregiving adversity. Suggesting a possible link to later physical health, greater abundance of Lacnospiraceae has been associated with markers of healthy immune system function in adolescents and adults. So now in figure 3, we also found abundance of several taxa were prospectively associated with internalizing and GI symptoms at age 4. Some of these results are surprising. For instance, that higher abundance of taxa from the Blaudia genus was associated with greater GI symptoms. Because in the adult literature, lower abundance of Blaudia has been found in patients with Crohn's disease compared to healthy controls. However, a lot more developmental work is needed to understand these findings more fully. So in future work with this cohort, we will examine the relations among adversity exposure, gut microbiota composition, and health symptoms using longitudinal models and additional data on these children when they're older to learn whether the microbiota may function as part of a causal pathway linking adversity exposure to health across childhood. And ultimately, this research may inform novel interventions targeting the gut microbiota to promote mental and physical well-being for children exposed to early adversity.